So I want to expand a bit on the issue of, on the point of short-term plasticity, and I want to expand the range of conditions. First of all, in terms of phenomenology, I'm going to talk about responses in auditory cortex, but I include in short-term plasticity short-term changes that operationally occur during experimental sessions. So this is something on the order of up to 10 minutes. Naively, these would be changes that do not involve translation transcription, but may involve activation of enzymatic cascade. So it's a bit wider than the type of mechanism that Dean just described. Now, I want to make a number of points about these mechanisms. First of all, they strongly shape neuronal responses to sensory stimuli. So last week I showed these things in the context of the workshop. But here you see, for example, an adaptation study of neurons in mouse auditory cortex. We have two stimuli, okay, the blue stimuli and the blue stimulus and red stimulus. And these neurons respond to both stimuli, but if you play a lot of the blue, they adapt very fast, but they keep the responses to the red stimuli. Okay, these are two pure tones that are very close in frequency to each other. So next, okay, they are extremely sensitive to the structure of the sequences. So, for example, in this experiment, we play these type of sequences, two-tone sequences, but in one condition, we organize the stimuli in a periodic pattern, and in the other one, they were random, with the same probability of the two tones. And you see significant differences in the responses. Okay, the more periodic, the more regular stimulus sequence give rise to smaller responses. These are intercellularly recorded sensory EPSPs in red auditory cortex. I'll skip this example, but it is also, it has the same character. And finally, these things have multiple time scales, which goes to at least hundreds of seconds. So I'll show you one example. So here again, we use sequences of two stimuli, complex stimuli in this case, and they differ in their internal structure, but all of both stimuli here are wideband. But we present always the two stimuli in two sequences. In one sequence, in this case, the TC10 is common and the TC3 is rare. And then later, we present the second sequence in which we switch the role. What you see here are the responses to the stimuli that act like TC10. So they're common in the first block and rare in the second. They adapt during the first block, and this adaptation is actually kept during the second one. The stimulus that was rare in the first block evoke response that's high in the first block and then adapts. So what you see here is, if you want a crossover of the adaptation from the first block into the second, and things here are hundreds of milliseconds, hundreds of seconds. Okay, so this is an adaptation that lasts a really long time. It develops over a few tens of seconds, but then it goes on for a long time. So the implications are, as I said, short-term mechanisms shape powerfully sensory responses. So everything that has to do with theta rate, stimulation, and so on and so forth will be strongly affected by these effects. Okay, long-term mechanisms, LTP induction, all of these things that we heard in the last 2 days operate on the landscape that is shaped by these mechanisms. And finally, there's no clear separation of timescales. This is for the theoreticians between us. Okay, basically, this short-term mechanism interpolate between membrane potential time constants and the many minutes that takes for long-term effect to occur. So that was the point that I wanted to make.
Okay, great. Let's open it up for a general discussion.
Something special about the mammalian auditory system for time, for interval discrimination, for short-term plasticity, because the auditory systems kind of organized around time. I mean, you mentioned cross-modal transfer of interval discrimination, which is really interesting. But is the auditory system just better at this than, say, the visual system? And sort of what mechanistically would account for the differences?
Based on discrimination, it's just fact. Yeah. Your auditory threshold for 100 milliseconds is probably 10% in the auditory system, but 20-25% in the visual system. So now sensory is pretty close to auditory.
Well, what do you mean by percent?
Percent. So if you have a 100 millisecond interval, your threshold is plus or minus 10 milliseconds. Whereas for the visual system, it's 100 milliseconds. Standard is plus or minus 25. So absolutely. Now why? I think it's probably a bit of both, meaning that, if I learn Morse code, I think my auditory cortex improves to do the temporal discriminations at those relevant intervals, which are incredibly complex. So I totally believe there's ongoing plasticity in all those systems. But I do suspect, as you know, there are architectural differences, particularly between B1 and A1. I'd love to know if the baseline short-term plasticity or the percentage of PB neurons and the different types of neurons. I don't know the answer to that. Does anybody know? If there's baseline differences between short-term plasticity in B1 and S1 or A1, or even percentage of PB neurons.
There definitely are differences in the architecture of S1 and B1. I don't know about A1, but we shouldn't assume that what we know about S1 is recapitulated identically in other parameters.
Can I actually come to the board? I want to present an alternative view of short-term plasticity that is different from what people seem to assign in terms of function to it. Usually people think it as adaptive filtering, right? So, if you think of a synapse, so this is presynaptic side and this is postsynaptic side. And okay, no, that's all the space I need. So what I'm going to suggest is that there is a feedback loop from the postsynaptic side to the presynaptic side through the neurons, okay, in the local circuit. And so the effect on the postsynaptic side of the EPSP, it may be sensed on the presynaptic side and affect the appearance of the spike because of that feedback loop. Then in the short-term plasticity in the paired pulse facilitation depression protocol, what you will get, you'll have two presynaptic spikes, and then the facilitation, say, to the second spike, in this view, is the result of this first EPSP going around and causing the second spike. At least that's what a synapse would think. Okay? And so then this facilitation will be a response to the probing of the loop with the synapse, and the synapse trying to kind of control the loop. The test of this idea would be to see if the potentiation, facilitation of depression is not just a function of the time lag between two spikes, but whether or not there was a first release. And it turns out, if you look at the experiments, actually the facilitation is a function of whether or not there is a first release. So there is some experimental evidence that there is this correlation. Anyway, just wanted to throw out this alternative view of what's going on in short-term plasticity. Marta?
In that case, if you just block the loop, you will lose the facilitation.
But the synapse doesn't know that the loop is blocked. So when people do experiments, depending on the preparation, there may not be a loop, but the synapse doesn't know. So it keeps acting as if it's controlling the loop. And so it's responding to these two spikes as if this is part of the feedback controller, right? Anyway, just wanted to put it out there.
brain is incredibly opportunistic, as is evolution, right? So I have no doubt that once a mechanism is in place, that these other additional potentially controlling mechanisms come into play. But I think it's also clear that there are many cases where short-term plasticity is really just doing adaptive filtering, in the sense that, so in the thalamocortical connection, there's no direct monosynaptic feedback loop. You'd probably argue, well, there's more complex feedback loops and so forth. But we know that there's beautiful data by Bruce Carlson in the electric fish, that they have these beautiful interval selective neurons, and those seem to be very feed-forward networks that are accounting for the interval selectivity that they observe. The same thing, data by Rose in the neuron system, in the frog system, where they have these beautiful interval selective neurons. And the responses are so quick that you don't really have time for much feedback. So I think in some cases, it's pretty clear that short-term plasticity is doing adaptive filtering and keeping a memory of the past. But I wouldn't dismiss this idea that in higher order areas that something like that might be happening, in my opinion.
But in thalamocortical, I mean, there's
feedback is just longer. Yeah, but it's just,
But short-term plasticity applies to the last of, tens of milliseconds, so there's plenty of time to get feedback from the cortex.
More questions? Oh, sorry.
Just wanted to raise something about the complexity, just of short-term plasticity without going into, and that is, so one of my former students actually has been looking at different synaptic tagments that there's 7 different isoforms, and some of them control fusion of vesicles, and of course, short-term depression is often thought to be, depletion of vesicles at synapses, facilitation. There's a sensor that sort of primes vesicles for release. But then some of the other processes are kind of layered on top of this, resupply of vesicles. And one of the synapses I study in the retina, there's a change. I'm not going to go into the detail, but there's a pathological change where the release probability drops a lot. So you see what used to be a depressing synapse turns into actually a facilitating synapse because the calcium current is reduced and there's less probability of release. But there's also a very dramatic change in the resupply of these vesicles because the synaptic tagment that's engaged in resupplying those, mobilizing vesicles so they can be delivered to the membrane, that doesn't work so well. And so you see this complicated change, which is almost an interval sensor, where there's no pair pulse depression, but if you stimulate above a certain frequency and above a certain number of pulses, you see the synapse just fall apart and no longer be able to release, and the recovery of that is slow. So I know that was a complicated scenario, but even short-term plasticity can give you, I think, more complicated timing rules that don't require long-term training of that short-term plasticity. And I don't know if people have taken that into account in the sort of thinking about the kind of things you've talked about, or is that baked in?
I think that's the standard view.
Okay.
That the sort of baseline short-term synaptic plasticity
is itself really complex.
Is already enough to inject temporal richness into the system. Okay. At least that's our standard view. I don't think there's that much focus. There's much focus on the computational function of short-term plasticity, but when there is, it's often in this adaptive filtering stuff. But in the network level, I would put it in the context of the reservoir. But building on what you say, there's a very good paper by Jackson and Regier, I think it is, on synaptotagmin 7. Jackman, thank you. In that paper, they show when they knock out synaptotagmin 7 specifically, that all short-term facilitation goes away at a number of different synapses. So the idea is that we have this absurd biochemical complexity, as Richard knows better than anybody, in the pre- and postsynaptic terminals, that seem to suggest to me that these things have the knobs to be switched, increasing synaptotagmin 7, decreasing synaptic 3, to precisely tune these things. So that's something that fits into our thinking. But
I think I didn't fully understand exactly what your prediction would be and why you drew a facilitation of this response to the second spike rather than depression. If you know, maybe I'm not fully understanding what the prediction would be for short-term plasticity that's creating temporal richness for doing temporal tasks versus what you might predict to see for short-term plasticity that's kind of allowing your synapse to act as a controller.
So I think, okay, so this doesn't predict whether it's depression or facilitation in the first place. I think that depends on the objective that synapse has as a controller. If it's trying to shut down the system or keep some level of activity, that would determine whether it's facilitation or depression. What I'm saying is that from the synaptic point of view, it may perceive the second spike as a causally influenced by its previous postsynaptic potential and change the magnitude of the next postsynaptic potential correspondingly. And the way to test it is to look at the second EPSP in those trials when you had a first or didn't have a first. And so if there is a correlation or anti-correlation in the magnitude like that, then this kind of is a supportive piece of evidence. And from the experimental data that
Measuring the loop for a given synapse to get feedback from its downstream.
the synapse is thinking that the second spike is somehow affected by its postsynaptic potential. So if there was a failure, then that spike would not be attributed to that. And then there would be less, say, facilitation. And in the data that I saw that there is that effect. I'm not trying to say that this is an exclusive role. The first spike from the synaptic perspective is not caused by its action at all, right? So it definitely came from the outside. So it is an adaptive filtering. I'm not trying to, it's not either or. I'm just saying that there is a component, I think, in STP which has to do with this feedback loop.
It seems like the second prediction from that idea is that something about the time constant for facilitation and depression would be related to the length of that, or the duration of that.
Exactly. Exactly. So that's the constructive part of this hypothesis, that you can use that time constant to start thinking which loop that synapse cares about.
I'd also like to ask other experts in terms of different time scales you have in the brain. There's these waves. They are not. They're just individual waves: alpha, beta, theta, and gamma, and all these. It's not kind of scaled versions of each other. Is there anything relevance? And these waves are often very global. They're not local. What are they playing in this time scale? Temporal discrimination, or love to hear.
Do you have any comments on the role of different oscillation in the type of auditory responses you see? I don't know much about.
Is auditory cortex more oscillatory than a visual cortex, for example? What is it? I like. Oh, you hate oscillations. I see. So, physiologically, is the auditory cortex more oscillatory than visual cortex or not? No? Less. Okay.
Not in our hands.
Okay, I see. Interesting.
really don't think there's much known about the role of oscillations, particularly in the temporal processing. There's a bit of data that suggests that when you create a temporal expectation, you know that the reward is coming in at 1 second, that there can be an entrainment of the slow waves, I think it's the delta waves, that entrains with the first stimulus. But I don't know much about it. Maybe somebody else here does.
You can create entrainment of delta wave in anesthetized animals and don't get any reward. So I don't know.
Because it's such a prevalent behavior, yeah, and it's always there unless the animal's dead. And you feel like, gosh, how can we put this in the story, yeah. And if you are very sleepy, the alpha wave gets strong, you know, aroused and so on. So.
Well, it's not that there's obviously activity at there is activity at delta rates, and there is activity. Yeah, but the question is to what extent this activity is related, driven, and trained, and so on by the sensory stimulation. I think that many of the studies that claim entrainment actually look at stimulus-driven responses that have been filtered through filters that have long time constants. And so it looks like entrainment, but in fact these are sensory responses. Okay? And so I'm not entirely sure about the interaction between sensory stimulation and the intrinsic oscillations.
So just the anecdote observations, I recently observed, in a visual task, the observers get quicker and quicker, and I always ask for their feedback, and then they say, oh, I feel more rhythmic. And I did not put any rhythm in my stimulus. They are spontaneously acting. Okay, they start a trial by pressing a button, they end a trial with their own thing, and then they can, make a sneeze and start the next trial. So there's no pacing. But nevertheless, it's this report of feeling rhythm. And then you see that at the same time learning to be faster and faster in the task. Of course, it's only anecdotal, but yeah.
Long-term plasticity of short-term plasticity. How do you think it operates? Do you have mechanistic insights into that?
No. No, I mean, I think, so sort of along the lines we were just saying, you could imagine things like synaptotagmin 7, that if the pairing happens late or early, and that maybe in addition to changing AMPA receptors, if the pairing happens late or early, you increase synaptotagmin 7 or not. But there is something very interesting, because I showed, and this I think relates to one of the questions that came up, it might have been Rob. The interaction between short-term plasticity and long-term plasticity typically has been fairly well established that when it occurs, LTP increases short-term depression, because the idea is that you're releasing more. There's this old paper that shows that when you induce LTP, this is actually in CA3 though, you induce LTP, the LTP goes up, paired pulse depression goes down. So I'm sorry, paired pulse depression gets more depressed. That's a bit, okay? So, which is everybody expects. But after you wait 3 hours, the LTP stays the same, but the short-term plasticity goes back to normal. So there's this idea that when we're just looking at the first half hour, the non-protein synthesis dependent early LTP, if you will, that's not the full story, because it might be the case that when you start looking at this is why when we're doing these experiments, we really think they have to be done in organotypic cultures in which you can follow the synapses over days or something. So, but to answer your question, Cliff, no, I mean, I think these are wide open questions, and yeah.
All right, this seems like an opportune time for me to ask my question. So I'm curious about whether you all feel, and I'll include Ellie in this, that the way that these concepts of metaplasticity are appearing or being studied in the hippocampus how well these concepts generalize with comparing to the auditory system and the hippocampal system. I was thinking during the first two talks that a lot of the concepts that were being raised had parallels in the auditory system, which I work on, that were called by different names. And then that's come out, I think, a bit in your talk, Dean, and then in what Ellie said as well. So are we all looking at the same phenomena, the kind of long history of work in the hippocampus, and then a kind of parallel work in the sensory cortex, and particularly in the auditory system where timing's always been so paramount? Are these the same metaplasticity phenomena, or are there special things? I don't know enough about the hippocampus to know if there are core differences either related to the area and to, the area being archaecortex cortex versus neocortex, or something else.
I don't know much about the hippocampus, and I know even less about the auditory system, so I can't really answer. I mean, the only thing I wanted to say is that these, in terms of GABA receptors, there are 3 of these extrasynaptic GABA receptor isoforms, alpha 4, 5, and 6. We were just talking about alpha 6, which is exclusively in cerebellum, I think. And alpha 4 is all over the cortex, and maybe they have similar. It hasn't really been looked at whether they migrate from extrasynaptic to synaptic locations.
I know about as much as of the auditory cortex as you. I guess my main thought here is that what I tried to get across at the start was that there's many types of metaplasticity phenomena. So it would not be at all surprising to find at least some of them that people describe in the hippocampus elsewhere. In fact, it'd be very surprising if they didn't. And it also wouldn't be surprising to me if people were calling them by different names as well, just like they do in the modeling, for example. They have different names for it. But I do think that metaplasticity is fundamental to the way a nervous system works in terms of, controlling plasticity. So I'm a biased observer, but that's my view.
When we have our computational hats on, we all hope, and it's probably true for most computation people here, you hope there's simple rules that you can use, incorporate into your models that capture cortical function or hippocampal function everywhere, right? That there's only one type of inhibitory, that there's only one type of STDP, that there's only one type of homeostatic plasticity or homeostatic. And, as I've grown older, I get more and more frustrated with the diversity of the biological systems we're studying. It's just so annoying because there's no hope to incorporate all of that into the models, right? Because so I fundamentally believe that there's different learning rules in the hippocampus and neocortex and different forms of metaplasticity. And that's why I put that slide up of the interaction between LTP and STP is just totally different in the neocortex and hippocampus. And it's probably different in different neocortical areas. Whenever we have these meetings, I get very depressed about diversity.
Yeah, so I completely agree, and I want also to reiterate what Alison said about the differences between V1, S1, A1. So that might also be expressed in learning rules. But I would also, part of the reason that I presented this short-term plasticity perspective is to urge you, the cellular physiologists, to put more emphasis on them, because we have accumulation of information across these hundreds of milliseconds of the type of processes that you were studying, but also over tens of seconds and over hundreds of seconds. There must be mechanisms, biochemical mechanisms that eventually implement this type of information accumulation. And so I would very much love to see these also studied, and not only learning rules of LTP and things like that. I think these short-term plastic learning rules or elastic changes type learning rules are important and understudied.
Okay. Any more questions? Oh.
I just want to address this question of feeling slightly overwhelmed by the biology. The question, and of course I'm, committed to understanding the details, but the question is what's the right level of abstraction? And so, it's appealing to think about nodes of the network and individual synapses, and in fact that might be how, the biological system operates. But I guess the question for you guys is, do we need that level of detail? Like if you can abstract away those short-term dynamics, can you still get a model that will be effective?
Get the NIH to fund more research into this area. So tell my study section.
I'm sorry, what am I supposed to tell them?
Tell them that we need to know more about what the cell molecular mechanisms are that are underlying second, tens of second, hundreds of second changes in operation of synapses and even intrinsic properties of neurons. Unless you don't want to know that.
We were chatting a bit about this at the coffee break earlier, and maybe I'm misrepresenting what you said, Steve, but I mean, one idea was we have all of these biological processes that look like they could be important computationally, but we don't actually have functions for them, like neural functions, like mapping these things onto actual computations that the brain is performing seems like, a big hole, right? Like we have it, we don't have a full description of all the cellular phenomena, but, we have a lot of phenomena, and what we don't have is the mapping onto what are they doing, and what multitude of computations might they be contributing to under what conditions. So, to me, I would like to know what all of the ones we know about are doing before funding more, putting more tools in the toolbox. I would like to know how the tools we have are used by the brain.
Okay. Why don't we stop there and continue the conversations over lunch? And let's give our speakers a... Yeah, after lunch, the afternoon is free, and there's beautiful hiking in the area. There's wine tasting that I know some of the people who are here, you can grab either staff or people who are here for the last few weeks to learn about that. And then an easy one is if you just walk to Campus Point down along the ocean here, you can rent kayaks and stand-up paddleboards and wetsuits and surfboards. And there's a nice beach there and a nice walk all along the ocean front. So have fun and see you tomorrow morning.